As you notice, we're going to be talking about the cell cycle and cell division in this lesson. Take a look at this photo and notice the two cells that are here. And then let's take a look at the inquiry question. Time to split. Unicellular organisms such as these reproduce when one cell divides into two new cells. The two cells are identical to each other. What do you think happens to the contents of the original cell before it divides? Okay, so by the end of the lesson, at least, you'll probably know that information. But think about it. What do you think? What do you think happens to the contents of the original cell before it divides? Well, I can tell you that the cells that you're seeing right here are paramecium, and they are found mostly in freshwater environments where they feed on bacteria. If you kind of take a look a little bit along here and a little bit along the edges here, you can see a hairy appearance of the dividing cell, and that's due to the cilia that covers its surface. The cilia are used for locomotion or movement and gathering food. Unicellular organisms such as these parameciums reproduce when one cell divides into two cells. Okay? We're also going to be talking about a couple of things, and they are in your vocab words, which are these here. So remember, you are going to be noting in your science journal all of those vocab words. You're writing them down. Okay? So we're going to start with cell cycle and interphase, sister chromatid, centromere, mitosis, cytokinesis, and daughter cells. So those are your vocab words that you need to note in your science journal. Also, please, let's write down these two questions, the essential questions. By the end of the lesson, you should be able to answer those. The first one, what are the phases of the cell cycle? Okay, we'll be going through that in this lesson. The second one, why is the result of the cell cycle important? All right, so those two questions need to be also written down in your science journal, and by the end of the lesson, you will be able to answer those questions. Okay, so at this point, I want you to stop the video and I want you to look at this question and this question, want them written down in your science journal, and I want you answering them to the best of your ability. What do you think happened to the contents of the original cell before it divides? That was the question that we asked down here. It's also here. I want you to write that information down. I want you to also answer that, please. The next one is this one here. Do you think these cells are at the beginning or at the end of cell division? The beginning or the end. The two cells here. The beginning or the end of cell division. And then it asks you to explain your answer.
The cell cycle. No matter where you live, you have probably noticed that the weather changes in a regular pattern each year. Some areas experience 4 seasons: winter, spring, summer, and fall. In other parts of the world, there are only 2 seasons: rainy and dry. As seasons change, temperature, precipitation, and the number of hours of sunlight vary in a regular cycle. These changes can affect the life cycles of organisms such as trees. Notice how the tree in Figure 1 changes with the seasons. Like changing seasons or the growth of trees, cells go through cycles. Most cells in an organism go through a cycle of growth, development, and division called the cell cycle. Through the cell cycle, organisms grow, develop, replace old or damaged cells, and produce new cells.
Okay, so you may have noticed in all of that, you had a vocab word. Please make sure you're writing the vocab word and its definition in your science journal. If you need to, stop the video, write the information down, and restart it again. Okay, so comparing cell cycle. Most cells in an organism go through a cycle of growth, development, and division called a cell cycle. Same way as a tree changes through the seasons. Through the cell cycle, organisms grow, they develop, replace old or damaged cells, and they reproduce or produce new cells. Okay, so at this point I want to ask you 3 questions, but today you get to pick which ones you're answering. Okay, so I'm going to make this a little bit larger. All right. What is a cell cycle? That's question 1. Number 2, what effect does the cell cycle have on an organism? And the third one, during spring, what part of a maple tree would have a high level of cell reproduction? So I want you to pick 2 of the 3. Pick 2 of the 3 to answer and write in your science journal. Okay? And then we'll move on.
Phases of the cell cycle. There are 2 main phases in the cell cycle, interphase and the mitotic phase. Interphase is the period during the cell cycle of a cell's growth and development. A cell spends most of its life in interphase, as shown in Figure 2. During interphase, most cells go through 3 stages: rapid growth and replication, or copying, of the membrane-bound structures called organelles; copying of DNA, the genetic information in a cell; and preparation for cell division. Interphase is followed by a shorter period of the cell cycle known as the mitotic phase. A cell reproduces during this phase. The mitotic phase has 2 stages, as illustrated in Figure 2. The nucleus divides in the first stage, and the cell's fluid, called the cytoplasm, divides in the second stage. The mitotic phase creates 2 new identical cells. At the end of this phase, the original cell no longer exists. Check. What are the 2 main phases of the cell cycle?
Okay, let's quickly look at Figure 2. That would be this figure here. Okay, I know this looks a little bit confusing, so we're going to walk through it fairly quick. Okay, this right here is the start. That is our cell, and that's where we're starting, is here. You'll notice that the arrows are going this direction, so this is the movement of the phase, or the information in the phase that we're looking at. Okay, the visual check question that was asked was, which stage of interphase is the longest? Well, if you take a look at our pie chart, which is what we're looking at as a pie chart here, you'll see that the dark blue segment, which is this one here, would be rapid growth and replication of organelles, seems to be the largest. Okay, seems to be the largest. As we move along, the next part to this cell, moving along to here, would be replication of DNA. Okay, and it only takes about half as much time as it does with the rapid growth and replication of organelles. As we move along a little further, you still see that we're in the blue, but it's also a shorter part to this, and that cell would be over here now in this information. Preparation for cell division. So it's preparing itself for cell division. Okay? Once we get to this part right here, you'll notice that it says mitotic phase. Right here. There's a light information there, as well as the yellowish or goldish arrows. Okay, so the cell division starts here with mitosis. Okay, mitosis, and then you see cytokinesis, which produces 2 equal identical cells. Now, I know it sounds confusing because we've yet to talk about mitosis, we've yet to talk about cytokinesis, but this chart shows you interphase and how long a cell stays in interphase before it starts to produce a second cell identical to itself. Okay, so at this point, we're going to back this down. All right, I want you to make sure that you've written down your definition for interphase. Interphase is the period during the cell cycle of a cell's growth and development. Okay, it's the period during the cell cycle of a growth and development of that cell. All right. Next thing, the asking of the questions. Okay, you get to again pick two, but one of those questions has to be this one. What are the two main phases of the cell cycle? You'll notice there's a gold key with a check mark. We need to make sure we know that one. All right, and then you can pick one of these two to answer also. You're writing these in your science journal and answering them. What happens during interphase? That one there. And the second one is this one. Why is it important that organelles are replicated during interphase? Okay, so you're picking one of these two and you're writing that one. All right?
Length of a cell cycle. The time it takes a cell to complete the cell cycle depends on the type of cell that is dividing. Recall that a eukaryotic cell has membrane-bound organelles, including a nucleus. For some eukaryotic cells, the cell cycle might last only 8 minutes. For other cells, the cycle might take as long as 1 year. Most dividing human cells normally complete the cell cycle in about 24 hours. As illustrated in Figure 3, the cells of some organisms divide very quickly.
Okay, so let's take a look at Figure 3 real quick. You'll notice that there's 4 pictures. This is 2-cell stage. This is a 4-cell stage. Notice 2, where it's starting to divide to 4. This is 32 stage, and this is 256 cell stage. This figure that you're seeing here is fertilized eggs of a zebrafish, and they divide into this stage, which is 256 cells, in about 2.5 hours. So it goes from this to that in about 2.5 hours. However, as was stated in the reading, most dividing human cells normally complete the cell cycle in about 24 hours. So all the cells are different. They're not the same. So when we talk about the cell cycle and how long it stays, how long the length of the cell cycle is, it varies depending on the type of cell. Remember also that only eukaryotic cells undergo mitosis. Again, the eukaryotic cells, that's a cell with a membrane-bound structure. Okay, we talked about that in a previous lesson, so you might want to review your notes so that you know. And the length of the cell cycle varies depending on the type of cell. All right, so now what I want to do is I want to offer up some questions. That would be these here. Okay. You get to pick one. Pick one question to answer, writing it in your science journal, please. Identify the type of cell that has membrane-bound organelles. Number 2 here. Summarize how the length of the cell cycle can differ among cell types. Number 3. Is it possible for different cells within a multi-cell organism to have cell cycles of different lengths? Okay, so now you're going to pick one of these 3 to write down in your science journal and answer the question, please.
Interphase. As you have read, interphase makes up most of the cell cycle. Newly produced cells begin interphase with a period of rapid growth. The cell gets bigger. This is followed by cellular activities such as making proteins. Next, actively dividing cells make copies of their DNA and prepare for cell division. During interphase, the DNA is called chromatin. Chromatin is long, thin strands of DNA, as shown in Figure 4. When scientists dye a cell in interphase, the nucleus looks like a plate of spaghetti. This is because the nucleus contains many strands of chromatin tangled together.
Okay, so the picture you're seeing here is a blow-up of the nucleus that's there. And if you look at those dark pieces of information in there, that is the chromatids. Okay? That is a chromatid, which is what was talking about just a second ago. Interphase. Okay, next. For you to understand each phase of the cell cycle, you must know about the important cell functions that prepare the cell for mitosis and take place during interphase. So what I want you to do is, I want you to pick one of these three questions to write in your science journal, okay, and answer them, and so we can talk a little bit more about it back in class. The first one, how is the cell's DNA referred to during interphase? How is the cell's DNA referred to during interphase? Question number 2, why might chromatid be tangled in the nucleus? Why might chromatid be tangled in the nucleus? And the third one, why might it be important that the cell make more protein during a certain part of the interphase? Okay, you're picking one of those three to answer in your science journal.
Phases of interphase. Scientists divide interphase into 3 stages, as shown in Table 1. Interphase begins with a period of rapid growth, the G1 stage. This stage lasts longer than other stages of the cell cycle. During G1, a cell grows and carries out its normal cell functions. For example, during G1, cells that line your stomach make enzymes that help digest your food. Although most cells continue the cell cycle, some cells stop the cell cycle at this point. For example, mature nerve cells in your brain remain in G1 and do not divide again. During the second stage of interphase, the S stage, a cell continues to grow and copies its DNA. There are now identical strands of DNA. These identical strands of DNA ensure that each new cell gets a copy of the original cell's genetic information. Each strand of DNA coils up and forms a chromosome. Identical chromosomes join together. The cell's DNA is now arranged as pairs of identical chromosomes. Each pair is called a duplicated chromosome. 2 identical chromosomes, called sister chromatids, make up a duplicated chromosome, as shown in Figure 5. Notice that the sister chromatids are held together by a structure called the centromere. The final stage of interphase, the G2 stage, is another period of growth and the final preparation for mitosis. A cell uses energy copying DNA during the S stage. During G2, the cell stores energy that will be used during the mitotic phase of the cell cycle. Reading check. Describe what happens in the G2 phase.
Okay, so you may have noticed I brought forward that same chart we were using earlier. That was this one, showing you a little bit more information using this chart or this table and this figure, showing how they connect, because here is your interphase, which is this in blue, okay, and the stages that it goes through, which is G1, G1, S and S, G2 and G2. You'll also notice mitotic phase, mitosis, mitosis is right here, and cytokinesis, which is right here, the 2 lines, and then the division of the cell into identical twos. Now, what I want you to do is, since I've shown you all that, and actually I'll show you a little bit more here, because we did refer to this figure also. The coiled DNA forms a duplicate chromosome made of 2 sister chromosomes. So here's one, and here's the other, and those 2 sister chromosomes are held together in the middle by a centromere. So it's like having 2 lines with something tying them in the middle. Okay? Okay, make that smaller, move this over, move this over. You had a couple of definitions that we need to go through, and I want you to write them in your science journal, please. The first one is sister chromatid, which is 2 identical chromosomes. They make up a duplicate chromosome. So 2 identical chromosomes that make up a duplicate chromosome. Okay, the second word is centromere, and sister chromatids are held together by a structure called a centromere. Those two terms, make sure you're writing them in your science journal. All right, so if you have to pause, please pause and then write them and then restart so that we can finish our lesson. All right, so I'm going to make that smaller. I'm bringing up our questions that I want you to answer or I want you to take a look at. Actually, I'll move that down here, make these a little larger for you. And you are to pick two of the four. Two of those four questions to answer, to write in your science journal and answer. What is the function of the centromere? Question 2, describe what happens in the G2 phase. Question number 3, how do chromatids and chromosomes relate to one another? And this one here, number 4, why would a mature cell that is no longer replicating remain in phase 1? Okay, or the G1 phase, phase 1. So you're picking two of these four questions to answer.
Organelle replication. During cell division, the organelles in a cell are distributed between the two new cells. Before a cell divides, it makes a copy of each organelle. This enables the two new cells to function properly. Some organelles, such as the energy-processing mitochondria and chloroplasts, have their own DNA. These organelles can make copies of themselves on their own, as shown in Figure 6. A cell produces other organelles from materials such as proteins and lipids. A cell makes these materials using the information contained in the DNA inside the nucleus. Organelles are copied during all stages of interphase.
Okay, so I need you to know that an important part of interphase includes the replication of organelles. Now you'll remember from a prior lesson that an organelle is a membrane-surrounded component of a eukaryotic cell with a specialized function or a specialized job. So what this is doing is it is taking those jobs, replicating them for the other cell or duplicating them for the other cell, and making sure that they are exactly identical. You'll notice in the picture, these are absolutely two identical cells. Notice they all have some of the same parts, okay, and you'll see the division right here. So to give you some additional information, you may not know that it is possible, okay, it is very, very possible, some organelles will replicate without involving the cell's nuclear DNA. So it won't involve the DNA at all. All it is the jobs that it's doing. So it's replicating those jobs. All right, so the next thing I want you to do is to write one of these three questions. One of these three in your science journal and answer it. At what point in interphase are the organelles copied? Okay, the second one, how do mitochondria and chloroplasts differ from other organelles in the cells? And the third one, what might happen to the daughter cells if a parent cell were exposed to a chemical that stopped its mitochondria from replicating? So pick one of those three to write down in your science journal and attempt to answer it from the information that we've given you.
The mitotic phase. The mitotic phase of the cell cycle follows interphase. It consists of two stages, mitosis and cytokinesis. In mitosis, the nucleus and its contents divide. In cytokinesis, the cytoplasm and its contents divide. Daughter cells are the two new cells that result from mitosis and cytokinesis. During mitosis, the contents of the nucleus divide, forming two identical nuclei. The sister chromatids of the duplicated chromosomes separate from each other. This gives each daughter cell the same genetic information. For example, a cell that has 10 duplicated chromosomes actually has 20 chromatids. When the cell divides, Each daughter cell will have 10 different chromatids. Chromatids are now called chromosomes. In cytokinesis, the cytoplasm divides and forms the 2 new daughter cells. Organelles that were made during interphase are divided between the daughter cells.
Okay, so now it's time to write your 3 definitions, that would be these 3 here, in your science journal, okay? Mitosis, the nucleus and its contents divide. In cytokinesis, the cytoplasm and its contents divide. And the third one is daughter cells. The daughter cells are the 2 new cells that result from mitosis and cytokinesis. Okay, so please write those. Make sure you're pausing and writing the information down and making sure that you have the definition correct, because we will be having a vocab quiz over that information. So make sure you have it right. Okay? Okay, so then the last thing I want you to do is this. You're picking one of these 3 questions, writing it in your science journal, and answering it, please. What are the 2 stages of mitotic phase? What are the 2 phases of the mitotic phase? The next one, what happens to the sister chromatids during mitosis? And the third one, a classmate says that mitosis occurs in bacteria. Is she correct? Explain your answer. So you're picking one of these 3 questions, please, and writing it in your science journal and answering it.
Phases of mitosis. Like interphase, mitosis is a continuous process that scientists divide into different phases, as shown in Figure 7, prophase. During the first phase of mitosis, called prophase, the copied chromatin coils together tightly. The coils form visible duplicated chromosomes. The nucleolus disappears, and the nuclear membrane breaks down. Structures called spindle fibers form in the cytoplasm. Metaphase. During metaphase, the spindle fibers pull and push the duplicated chromosomes to the middle of the cell. Notice in Figure 7 that the chromosomes line up along the middle of the cell. This arrangement ensures that each new cell will receive 1 copy of each chromosome. It is important that new cells be identical. Incorrect genetic information in the new cell can lead to the uncontrolled division and growth of new cells, known as cancer.
Okay, so just like interphase, mitosis is also divided into phases. The first 2 phases of mitosis appear simple, but they are important because they ensure that the new cells receive the complete sets of information they require. Okay, so take a look at this information right here. You'll notice prophase and metaphase. We're going to have 2 diagrams. This is the first one showing the first 2 phases. It gives you this diagram as well as a real cell. This is it pulling everything together and spindles in the middle. Okay, the metaphase is here. You'll notice the pulling here of the chromosomes to the center. And if you notice the actual cell, you'll see the dark spots in the middle right here. That is this process going on. Okay, so those 2 things are quite important, even though they seem really simple. Okay, so I'm closing that down for a second. If you want to take a look a little harder at it, then make sure that you pause the video and really view that diagram. Okay, so in this case, I want to bring this information up. Okay, moving that one over there. Okay, I want you to write down all 3 of these. These are all 3 very important questions, and I want you to answer them to the best of your ability. So question number 1, what are the phases of mitosis? Okay, question number 2, what structures appear and disappear during prophase? And question number 3, what important step takes place during metaphase? And why is metaphase so important? So you're writing all 3 of these questions in your science journal and you're answering them.
Anaphase. In anaphase, the 3rd stage of mitosis, the 2 sister chromatids in each chromosome separate from each other. The spindle fibers pull them in opposite directions. Once separated, the chromatids are now two identical single-stranded chromosomes. As they move to opposite sides of a cell, the cell begins to get longer. Anaphase is complete when the two identical sets of chromosomes are at opposite ends of a cell. Telophase. During telophase, the spindle fibers begin to disappear. Also, the chromosomes begin to uncoil. A nuclear membrane forms around each set of chromosomes at either end of the cell. This forms two new identical nuclei. Telophase is the final stage of mitosis. It is often described as the reverse of prophase because many of the processes that occur during prophase are reversed during telophase. Reading check. What are the phases of mitosis?
Okay, so anaphase and telophase are the last two phases of mitosis. Okay, it may be hard for you to understand what goes where in these two phases, but just remember that prophase is the opposite from telophase. So when we start, the process is almost the exact opposite from what's going on between those two phases. Okay, so take a quick look again. Pause the video and look, and take a look at both diagrams that I had just given you. This one here with these two phases, and the other one showing the first two phases, okay, which is prophase and metaphase. Those two diagrams are kind of important because it gives you a big overall picture of what this is all about. Okay, let me go ahead and close this one, move this one over. All right. Now, pick one of the following three questions to answer in your science journal, writing the question and the answer, please. First one, give reasons why telophase is the opposite of prophase. Question number 2, what might happen if the spindle fibers fail to shorten during anaphase? And question number 3, what would happen during mitosis if the chromosomes did not replicate during interphase? So one of those three questions, you are writing it down and answering it in your science journal.
Dividing the cell's components. Following the last phase of mitosis, a cell's cytoplasm divides in a process called cytokinesis. The specific steps of cytokinesis differ, depending on the type of cell that is dividing. In animal cells, the cell membrane contracts or squeezes together around the middle of the cell. Fibers around the center of the cell pull together. This forms a crease, called a furrow, in the middle of the cell. The furrow gets deeper and deeper until the cell membrane comes together and divides the cell. An animal cell undergoing cytokinesis is shown in Figure 8. Cytokinesis in plants happens in a different way. As shown in Figure 8, a new cell wall forms in the middle of a plant cell. First, organelles called vesicles join together to form a membrane-bound disc called a cell plate. Then the cell plate grows outward toward the cell wall until two new cells form. Reading check. Compare cytokinesis in plant and animal cells.
Okay, well, as you take a look at Figure 8, which is this one here, there is a difference between animal cells, which is this one on the left, and plant cells, which is that one on the right. The one on the left actually is without a cell wall, if you notice. Here's your cell wall. Okay, so this one is distinguished as an animal cell. And the membrane on the outside pulls that direction, okay? And this one will pull the other direction, leaving this furrow in the middle, which means it's opening up an area where it's going to pull apart to form two separate cells. Okay? So that in itself, this one is different than this one. Two different types of cells, two different ways of cytokinesis. Okay, so now, make this smaller since we just talked about it. And you need to pick one of these two, please, to answer. Pick one, write it in your science journal, and answer it. Would a cell plate form in your own body during cytokinesis? Would a cell plate form in your own body cell during cytokinesis? And it asks you to explain. Please do that one or this one here. Compare cytokinesis in plant and animal cells. Okay? So you're picking one of those two questions to answer.
Results of cell division. Recall that the cell cycle results in two new cells. These daughter cells are genetically identical to each other and to the original cell that no longer exists. For example, a human cell has 46 chromosomes. When that cell divides, it will produce two new cells with 46 chromosomes each. The cell cycle is important for reproduction in some organisms, growth in multicellular organisms, replacement of worn-out or damaged cells, and repair of damaged tissues.
Okay, so now what I want you to do is to pick one of these three questions to write in your science journal dealing with the results of cell division. Okay, you can do this one. How many chromosomes does a human have in each body cell? Or this one. Identify two reasons the body needs to make new cells. Or number 3, why does an original cell that undergoes mitosis no longer exist? So you're picking one of those three to write the question down in your science journal and answer it, please. Okay, so the next information we're going to cover fairly quick, okay? Because I know it's your most favorite thing of all in science, and that is the math. Okay? We do need to use math in science, and in this case, we do use percentages in science. Okay, so now we're going to quickly look at this information here. Use percentages. A percentage is a ratio that compares a number to 100. If the length of the entire cell cycle is 24 hours, 24 hours equals 100%. If part of the cell cycle takes 6 hours, it can be expressed as 6 hours divided by 24 hours. To calculate percentage, you divide and multiply by 100. So in this case, you're taking the 6 hours, which was the part of the completed cell cycle, divided by the total amount, which was 24. So you're dividing 6 by 24, which gives you 0.25. That 0.25 is then multiplied by 100 to give you 25%. Okay, so let's practice this. In your science journal, I want you to write this practice question down. Okay, I want you to write the equation, not the practice question, but I do want you to write the equation. Interphase in human cells takes 23 hours. If the cell cycle is 24 hours, what percentage is interphase? So you're going to do the math. Here's your example. Do the math, get an answer, and we'll talk about it in class.
Reproduction. In some unicellular organisms, cell division is a form of asexual reproduction. For example, an organism called a paramecium often reproduces by dividing into two new daughter cells or two new paramecia. Cell division is also important in other methods of reproduction in which the offspring are identical to the parent organism. Growth. Cell division allows multicellular organisms, such as humans, to grow and develop from one cell, a fertilized egg. In humans, cell division begins about 24 hours after fertilization and continues rapidly during the first few years of life. It is likely that during the next few years, you will go through another period of rapid growth and development. This happens because cells divide and increase in number as you grow and develop. Replacement. Even after an organism is fully grown, cell division continues. It replaces cells that wear out or are damaged. The outermost layer of your skin is always rubbing or flaking off. A layer of cells below the skin surface is constantly dividing. This produces millions of new cells daily to replace the ones that are rubbed off. Repair. Cell division is also critical for repairing damage. When a bone breaks, cell division produces new bone cells that patch the broken pieces back together. Not all damage can be repaired, however, because not all cells continue to divide. Recall that mature nerve cells stop the cell cycle in interphase. For this reason, injuries to nerve cells often cause permanent damage. Check. Why is the result of the cell cycle important?
Okay, so the information just provided to you was reproduction, growth, replacement, and repair. I want to see if you understood any of the information that was provided to you just there. So I'm going to make this smaller and ask you 3 questions. You get to pick 2 questions, however, one of those 2 questions has to be this one. Okay? So you're picking one of these 2, and you're definitely writing this one down. Writing them down in your science journal, please, and answering those questions. Number 1, why is cell division important for repairing the body? The second one, which is one you have to write down because it's gold key check. Why is the result of the cell cycle important? And the third one, which may be one you choose, certain cells such as nerve cells remain in interphase after maturing. So what would happen to these cells if they were injured? So you're picking one of these 2 to write down in your science journal and answering the question, and then you're definitely writing this one down and answering it. Okay, so now you've come to the review section at the end of this lesson. I want you to answer the questions on the page. You do not have to write the question for this one, or this one. You do have to write the question for this one and only the answer that pertains to it. And you're going to be answering the other ones. Okay, you are drawing the chart, please. If you are confused as to what you're supposed to write down and what you're not supposed to write down, just write down all the questions except for these 3. Okay, you do not have to write these 3 questions down. You're also doing the math question, please. So, all of that in the science journal. Do your best, and I hope you understood the lesson. We'll be talking about it in class.